Hello, I am Dr. Shanmukh and I am here to discuss about the important yet a small topic in a very precise manner, the neural development and some of the defects of neural development. So what happens here? We are going to discuss very briefly and some concepts of how and why the nervous system is developing in the way that it is right now in an adult brain. So moving forward, we know around the third week of the embryonic life, the primitive streak after forming will form a primitive node and through which an invagination and migration of cells happen towards the anterior end, towards the buccal membrane, and this will get hollow with time. It becomes hollow to form a notochordal process, and this notochordal process is filled by the ectodermal cells in due course of time in the later stage of the embryonic life, and this will form a tube-like structure. It will form a tube-like structure. This is the notochordal process, and this notochordal process will be fusing with the endoderm and form a notochordal plate, and it will form a notochordal plate. This notochordal plate, again, what it does, it makes a tube-like structure and it folds, and then it proliferates completely to form a proper notochord. So this forms a notochord. This is the definitive notochord. This is the definitive notochord. Why is notochord formation is such an important step? It is because you can see here, it determines the axis, point number one, and it gives a medial and lateral directions in a developing embryo, and it functions as an inductor. Inducting what? Exactly what? The neural plate formation, differentiation of cells, and vertebral column formation. So this notochord produces multiple signals or multiple chemicals that induce all these. This is just a tip of iceberg of what notochord can do. As we know, notochord remnants in adult is nucleus pulposus of the vertebral column. As we see here, this notochord will send signals towards the ectoderm. Okay, so there is a very important point here. What happens when this is the embryo and this is the ectoderm and this is the endoderm? Usually, without some signals, the ectoderm is invariably is designed to develop into skin. It is invariably designed to develop into skin. Okay, this happens with the help of BMP, called as bone morphogenic protein. So bone morphogenic protein induces development of ectoderm into skin. So what happens? There are some factors produced. These are called NFC factors, that is noggin, folastin or folastatin, and cotin. These factors, they block the development of ectoderm into skin, and they induce development of neuroectoderm. Okay, so neuroectoderm development needs a special chemical. From here, this will cause differentiation of this ectodermal epithelium into columnar epithelium to form neuroectoderm. So this neuroectoderm, in meantime, it folds into an invagination. It now has a basal side, a basal side, a crest. This is the crest. Okay, this is the invagination, neural groove. We call it the neural groove. This neural groove, it will fuse, these edges will fuse together to form a neural tube. To form a neural tube. So what happens to the crest cells? What happens to them is they will lie on the superolateral side. They pinch off from this area and they lie on the superolateral side of the neural tube. So neural crest cells continue their work. Okay. So this BMP is initially produced by the ectoderm and it induces a skin formation. In presence of NFC, the neuroectoderm develops into neural tube and neural crest cells. This neural tube has dorsal plate and ventral plate. So here, as we have seen, this neural tube has a dorsal plate and a ventral plate. In spinal cord, we see that there is a development of sensory system is imbibed in the dorsal part of the spinal cord, and the ventral side it is the motor. So what happens here is very interesting actually. So dorsal plate and the ectoderm secretes a lot of BMP. So BMP on ectoderm will lead to skin formation. But once neuroectoderm is formed, the same BMP will induce the differentiation into sensory and interneurons on the dorsal side, because in the embryo, what is seen? This is the neural tube and this is the ectoderm and this is the endoderm and here is the notochord. We are seeing that in the neural tube, this dorsal is close to the ectoderm. So ectoderm and the dorsal plate both produce BMP, bone morphogenetic protein, which will induce the differentiation of this neural tube into sensory on the dorsal side. Okay, as on the ventral side, what is happening? This ventral plate and notochord, they secrete SHH. It is nothing but sonic hedgehog protein. So this acts on the ventral aspect of the neural tube and differentiate and develop into the motor neuron. So this is very important to understand. So here it says to the for the formation of neural crest cells, you should have intermediary signal of both BMP and as well as the sonic hedgehog protein. Okay, I hope you understood and I hope this is clear how this neural tube is developing into dorsal side of the spinal cord. And this neural tube itself has multiple dilatations and it will develop into forebrain, midbrain, hindbrain, etc. Right? So how is it possible? There is something called, there are some proteins or genes called HOX. This is the HOX gene actually. I should not call them protein. HOX genes. HOX genes, they determine from which part of the brain which structure comes out, like the cranial nerves 3 and 4 from the midbrain, 5 6 7 8 from the pons, 9 10 11 12 from the medulla. So all this is predetermined. So whatever that we are seeing, it is completely predetermined by the genes. The genetic factor plays a very important role. As well as in development, what factor plays a very important role is the environmental, that is both learning and the exposure, including the dietary, that is a nutrition factor. Okay, that helps in the formation of synapses and all. Coming to the cranial dystrophism, there is anencephaly, that is failure of development of the brain itself. Okay, here the folate deficiency takes over. Why folate is important means it helps in proliferation. When there is deficiency, there is decreased growth and differentiation and proliferation, because it helps in cell cycle and cell division. Okay, so there is no closure of neural tube and also there is no development of brain. So this leads to anencephaly. Okay, and we can see here multiple defects. The normal, how it is, the skin, dura, metal, abdomen, hinges, the bone, the vertebral column and all. and there is spina bifida occulta. Okay, and this will be seen with a tuft of hair with and a skin dimple, that is a depression. And meningocele, only the meninges are coming out of this defect. And mylo means the neuron, that is myelination. The myelinated structure is the spinal cord is also coming out. It is very bad condition, myelomeningocele. And coming to the brain malformations, holoprosencephaly, sonic hedgehog. This gene is responsible for uh, ventral midline patterning. So sonic hedgehog gene is very much responsible for neural tube closure as well as ventral midline patterning. This will be absent, and it entire thing appears as one without any division in the brain. Okay, that is why you can see even holoprosencephaly, cyclopia. That means you call it hypertelorism when there is more distance between both the eyes, that is lost here. So cyclopia, one eye is seen, and there may be associated nasal defects. Sometimes nose is absent. Okay, and we can see this will lead to holoprosencephaly. That means the fused brain, no differentiation. This is seen in Patau syndrome. Okay, and presence with midline defects, as we have seen. Single ventricle, as you can see here, the single ventricle. Fused basal ganglia, cleft lip, palate, hypotelorism, cyclopia, proboscis. Proboscis is absence of nose, and in place of nose, the eye, there is a tube like structure coming out of the eye, that we call proboscis. We see in the insects. Okay, lissencephaly. Listen means it's a smooth brain, no sulci, gyri, etc. So what happened? The brain cells develop near the ventricular surface. If this is the brain, if this is the brain, if there are ventricles here, okay, this is the developed ventricle, and during the development, this is the canal, neural canal. There is fluid inside, and the development happens towards this side, and these cells migrate towards the outward surface. Okay, this will lead to formation of sulci and gyri in the brain. Apart from the other genetic coding, this is also important factor, and this is governed by microtubule movement. It is dependent on microtubule movement, and this microtubule is coded by LIS1 gene. If this is defective, lissencephaly, that is the smooth brain. Okay, there are multiple grades of lissencephaly based on how much it is there. Uh, and the LIS1 is not the only gene which causes this defect, but it is one of the most important genes. And abnormal bone developments. Okay, this is seen in BMP signaling or any defect with the BMP protein. Any defect, abnormal bone development, because it is bone morphogenetic protein. Okay, posterior fossa malformations. It is Cherry 1 malformation or Arnold Cherry malformation, as we can it was used to be called as. Okay, usually this is the downward displacement of cerebellar tonsils through foramen magnum. So this is usually asymptomatic because only the tonsils are descended. Okay, this is Cherry 1 formation. Usually this is also associated with abnormal bone development, because if the skull development is impaired, all the skull plates, okay, you cannot expect the brain development to be normal, that it should be in place, because this is the bony vault which contains the cranial structures in place, and once this is defective, there are other malformations. So Cherry 2 malformation is displacement of cerebellum, the vermis and tonsils, and medulla structures, okay, through foramen magnum. So this will form non-communicable. This will block the canal from the behind because of the herniation, and it will lead to hydrocephalus. But the communication between the cranium and the spinal cord is lost. It is severed because of the displacement. Okay, it is more severe than cherry one, obviously, and it presents with dysphagia, stridor, apnea, limb weakness associated with myelomeningocele. Okay, myelomeningocele. So dandy walker malformation. This is agenesis of cerebellar vermis. Okay, this is agenesis of cerebellar vermis. This is cystic enlargement of fourth ventricle. Cerebellar vermis rise directly above the fourth ventricle, and if there is agenesis, you can see the dilatation as we see here. This is the cerebellum. You can see the arborations and see the malformation. How much? You can see the fluid in white here. This is because of FOXC1 gene defect. This is implied in cerebellar and hindbrain development. Okay, FOXC1 gene. And this abnormality will lead to Dandy-Walker malformations. Okay, and after this, we can also see how it folds into its own structures, and that we'll see in a minute. As we can see here, this is the prosencephalon, as we can see. And from the prosencephalon, then we have a connection to the mesencephalon, and then to the rhombencephalon and the spinal cord, as we can see. From the prosencephalon, we can see the optic vesicle development here. You can see the optic vesicle development here. It is initially developed laterally and then it comes forward with the development. Okay, and these things, the balloon-like structures that you can see here, these are the cranial and the spinal ganglia. Okay, and we can say, in the neural development, these adrenergic and cholinergic neuron, how they are developed is because of the neural crust cells. These neural crust cells, upon action of some mediators, they differentiate into adrenergic and cholinergic neurons. From there, we can see this forebrain. From the optic vesicle, we can see, and here is the diencephalon that is coming from the forebrain, and it is in connection with the mesencephalon. You can see how it is folding itself in the cranial cavity and how it is coming out as a spinal cord, and this will become the proper adult functional brain with the sulci and gyri. If this sulci and gyri are absent, it becomes lissencephaly. Okay, see how much Hox genes we have spoken. Hox genes, they play a important role in determining which part of the brain or anything comes from which segment. Okay, and this is the pontine flexure. Okay, in future this will form multiple structures. This is how the folding occurs, and this is the cross section. You can see prosencephalon, metencephalon, rhombencephalon, and the spinal cord. And here the prosencephalon is, this is the telencephalon, and this is diencephalon. Again, the mesencephalon remains the same. The optic vesicle develops from the diencephalon. As we can see here, the basal ganglia develop. This diencephalon, this one, remains underneath the cortex as thalamus and hypothalamus. Okay, we can see here what is developing from what. The telencephalon, it will give rise to the cerebral cortex. We have seen it, and the cerebral nuclei, that is the basal nuclei, the amygdala, and the basal forebrain, and it is associated with lateral ventricles, as we all know. And the diencephalon, this will give rise to thalamus, hypothalamus, and also retina, and it is associated with third ventricle. This is very, very important. Retina develops from the diencephalon, thalamus, and hypothalamus. We can see that diencephalon is a connection between mesencephalon and the cortex, right? So it should be a mediator, like between the rest of the brain and the cerebral cortex. So what is that relay station? That is thalamus for, you know, the sensory signals, and between all these basal nuclei pass the motor tracts. So this is very, very important juncture. Okay, and mesencephalon, that is the midbrain, superior inferior collicular red nucleus and substantia nigra. This develops from the mesencephalon, and the cerebral aqueduct is the cavity associated. Metencephalon and myelencephalon are a part of rhombencephalon. Rhombencephalon, metencephalon, the cerebellum and the pons. Okay, metencephalon defect, the worms cannot be formed. You have seen. Okay, and cerebral aqueduct, sorry, the fourth ventricle is the cavity of this metencephalon. Myelination, it's most of the times myelination is, we call it nerves periphery. So the medulla oblongata, which continues as spinal cord, that is the fourth ventricle, and the spinal cord is spinal cord and it has central canal. So this is how the development occurs. The interneurons are developed from the BMP signaling, and the motor neurons are developed from the sonic hedgehog signaling. And with optimum dosage of this BMP and sonic hedgehog, the neural crystals develop, which later migrate. Okay, these neural crystals NCC later migrate because of multiple signals from the somites. Okay, notochord, alar plate, and dorsal. So these are the inducers of CNS. Okay. So this neural crystals will migrate to multiple parts and form the melanocytes, the adrenal gland, okay, and even corticotropic cells and all. Okay. Thank you.